Hi class! In this lesson you will learn how cells reproduce by dividing. In the corner here is a picture of a cell that's undergoing division, and the DNA has been stained in blue and the spindle apparatus that separates the DNA is stained in green. It's just such a gorgeous photo. Biological processes can be so beautiful. Well, let's first look at the lesson overview. First, I'll just introduce you to when cells divide and the different types of cell division. Then we'll talk about how DNA is packaged into chromosomes. And then most of the lesson will focus on the stages of the cell cycle and a type of division called mitosis. And then at the end, just a brief introduction to how cells exit the cell cycle and a special type of division called binary fission, which occurs just in prokaryotic cells. First, I'd like to ask you, when do you think cells need to divide? For what purpose would a cell undergo cell division? Well, think about it. Hopefully you're thinking for growth. As an organism is growing, it needs to make new cells. Another purpose is for replacing any dead cells. You're actually constantly shedding dead skin cells, and you might sometimes even exfoliate to remove dead skin cells on purpose. Well, those need to be replaced through cell division, otherwise you'd soon run out of skin. And the third reason would be for reproduction. Cells need to divide when an organism needs to reproduce to make new offspring. There are two major types of cell division in eukaryotic cells. The first one is mitosis. This is the one that this lesson will focus on. Mitosis creates an exact copy of the parent cell, of the original cell. The two new cells that are created are exactly the same as the original. In multicellular organisms, it occurs in the somatic cells. Somatic cells are your body cells, everything except your sex cells. And some eukaryotes can even use mitosis to reproduce asexually. The second type of division is called meiosis. Meiosis has one purpose, and that is to produce gametes, which are egg and sperm. Prokaryotic cells, they do not undergo mitosis or meiosis. They have a different kind of division called binary fission. And prokaryotes are all single-celled organisms, so they use binary fission just for the purpose of asexual reproduction to make new prokaryotes. Now imagine that this cell is about to divide, so it's just going to get split into two new daughter cells. What does it need to accomplish? Well, it needs to make sure that the two new daughter cells have all of the organelles that they need. So the organelles need to be divided. Then even more tricky, the DNA inside the nucleus has to get properly divided. We need to make sure that the two new daughter cells have the same amount of DNA as the original cell, that they don't lose any of the genes that they need to carry out their cellular functions. In this lesson, we're going to focus on the division of the DNA, which is packaged into chromosomes. So here, let's first take a look at how DNA gets packaged into chromosomes. Chromosome is basically one DNA molecule that has been wrapped around certain proteins and packaged around the proteins to be a more condensed chromosome. So let's follow this diagram from the bottom. Here at the bottom, you just have the basic DNA double helix. Then up here, you can see that the DNA double helix gets wrapped around these little proteins called histones. So a histone is a protein that's pictured here as just one of these little circles. The DNA is wrapped around a group of histones, and this one unit of DNA wrapped around histones is called a nucleosome. So each of these, what look like little beads, each of them are nucleosomes. This DNA then gets coiled and then supercoiled into the fully condensed chromosome. When a cell is not dividing, the DNA doesn't need to be fully packaged. So when the cell's not dividing, it's found in the coiled form. But when the cell is about to undergo cell division, then it gets supercoiled into this chromosome. And that's so that the DNA is smaller and easier to move around in the cell. Here's just a simple diagram of a chromosome to help us learn some of the important parts. So here you have a single chromosome, And when the cell is going to divide, that DNA molecule needs to be duplicated. It's replicated into two attached DNA molecules so that when the cell divides, these two DNA molecules can be separated from each other so that the two new daughter cells each get the same DNA. Now for some parts. This constricted region where the two replicated DNA molecules are attached to each other, this is called the centromere. And these two attached DNA molecules are each called a sister chromatid. So these are the sister chromatids. Okay, so just a basic overview of what's going to happen to the chromosomes. Chromosomes get duplicated. Here you can see the little double chromosomes. Then they line up in the middle of the cell, and then those sister chromatids get pulled apart. Once they're pulled apart, each of these is considered to be a new individual chromosome, and they get separated into two new nuclei, and then the cell divides. And the two daughter cells end up with the same amount of DNA as in the original cell. Now let's take a look in more detail at the stages of the cell cycle and mitosis. The cell cycle is basically the life of a cell. It's all of the events from the birth of a cell as it's growing, carrying out its functions, until it divides, when each of the two new cells begin the cell cycle all over again. The cell cycle can be divided into two major phases. Interphase, this is the longest stage, shown here in orange. And then the mitotic phase, which is much shorter than interphase. This is when the cell is dividing and is shown here just in yellow. Each of these can then be subdivided into other stages. Interphase is subdivided into G1, S, and G2. Then the mitotic phase is subdivided into mitosis and cytokinesis. Mitosis is the division of just the chromosomes, and cytokinesis is the division of the cytoplasm. Now we'll take a look at the stages in more detail. So first, interphase. Remember, this is the longest stage. This is when the cell is not dividing. And the first step is G1, which also stands for gap 1 or first gap. This is when the cell is growing, it's making more cytoplasm, more organelles, and it's carrying out all of its metabolic functions. So whatever that cell has as its job, say if it's a cell in your pancreas and its job is to make digestive enzymes, that will be happening in G1. Then the S phase. The S phase stands for synthesis or DNA synthesis. This is when the cell is duplicating its DNA. And then G2 is gap 2. This is when the cell is growing more, carrying out more of its functions, and it's getting ready for cell division. So probably the most important part about interphase to remember is when the DNA duplication takes place. So DNA is replicated during the S phase, DNA synthesis. Now we get to the stages of the mitotic phase, which, just to remind you, is divided into two big parts: mitosis, which is the division of the chromosomes, and cytokinesis, the division of the cytoplasm. Then mitosis can be separated into four major phases: prophase, metaphase, anaphase, and telophase. The way you can remember the order of these stages is just remember PMAT. Okay, so prophase. A whole bunch of things happen in prophase. The nucleolus, that dark circle in the nucleus, this disappears. Chromatin fully condenses into chromosomes. The DNA gets supercoiled. And you get formation of the mitotic spindle, which is shown in blue here. This is where the chromosomes are going to attach themselves so that the sister chromatids can be separated. And then we have another stage which you don't need to remember this name. Just think of this as a continuation of prophase. More stuff happens, nuclear envelope disappears, and the chromosomes attach themselves to the microtubules. Now we move on to metaphase. Metaphase, easy to remember. Chromosomes simply align in the middle of the metaphase plate. It's what's sometimes called the equator of the cell. In anaphase, the sister chromatids separate from each other and begin to move towards opposite poles. And then telophase is when the chromosomes have now arrived at the two opposite poles of the cell. Two new nuclei begin to form, and the chromosomes uncoil partially so that they're now back into chromatin. The nucleoli appear in the two new nuclei, and the cells now continues to elongate to prepare itself for division of the cytoplasm. And here we have cytokinesis, the division of the cytoplasm, and we're done. So now let's just look at the summary. So we had prophase, metaphase when the chromosomes align at the equator, anaphase when the sister chromatids are separated from each other, telophase when the two new nuclei form, and then cytokinesis.